Welcome to MistyBiology, let's learn something new. Today I will discuss the Lac Operon, structural genes, and the gene regulation for the metabolism of lactose. Here the lac stands for lactose, which is a disaccharide sugar, and is used by the bacteria for energy source. Operon means, group of genes, under a single promoter. Here you can see, when the glucose is high, E. coli happily enjoys the glucose as its food. But if the glucose is absent, and lactose is present, it uses lactose as its energy source. It can still take the energy when its favorite food is not present. E. coli uses lactose for energy, when glucose is not present. So, there are some genes required for the metabolism of lactose in E. coli. For example, lacZ gene after transcription, produces beta-galactosidase enzyme, which breaks down the lactose. lacY helps to take up the lactose, and lacA gene produces beta-galactoside transacetylase, that transfers an acetyl group from acetyl-CoA to beta-galactosidase. Here, you can see, the enzyme beta-galactosidase encoded by lacZ gene, converts lactose into glucose, and galactose. In this slide, you can see, the enzyme permease encoded by lacA, which helps in the transport of lactose into the cell. Here, the permease acts as a transport molecule, for lactose. When Lac Operon is not transcribed, the number of lactose transport molecules will be negligible, that means, now no lactose can enter inside the cell. In the second case, when Lac Operon transcribes, it forms a greater number of permease, or transport molecule. Now more lactose can enter, and then metabolized by the E. coli. Thiogalactoside acetyltransferase transfers, acetyl group from acetyl-CoA to beta-galactoside. Though, the role of this enzyme remains unclear, but it was seen that, it plays a role in detoxification, by preventing acetylated galactosides, from re-entering the cell. So far, we have discussed about, what is Lac Operon, and the role of structural genes. Now, we will see how the regulation of the genes take place. Have you ever thought about, how a cell detects the level of glucose, and lactose? How it differentiates, between monosaccharide and disaccharide? E. coli senses the level of glucose, and galactose, with the help of two DNA-binding protein, Lac repressor, and catabolite activator, CAP, protein. Here, Lac repressor acts as a negative regulator, while CAP acts as a positive regulator of Lac Operon. Now, we will discuss about Lac repressor. The transcription of Lac Operon is repressed, by the Lac repressor protein. In order to accomplish this, it binds to the operator partially overlapping promoter. It senses allolactose, an isomer of lactose, and changes its shape. When bound, the Lac repressor gets in the way of RNA polymerase, and prevents operon transcription. When lactose is absent, repressor will bind directly to operator, and stops RNA polymerase to transcribe structural genes. Why would a cell waste energy to form enzyme if there is no lactose? When lactose is present, and glucose is absent, then, some of the lactose molecules will be converted into allolactose, which is the isoform of lactose. This allolactose changes shape of repressor molecule, which will not be able to bind to operator. Now, when operator sequence is free, RNA polymerase can easily move, and transcribe all the structural genes. Now, let's discuss about catabolite activator protein, which is a DNA-binding protein, and a positive regulator of Lac Operon. It helps RNA polymerase in the transcription. It senses glucose, and is regulated by small molecule cyclic AMP. Whenever the glucose is low in the environment, cyclic AMP will be high, and high transcription of structural genes occur. When the glucose is high, no cyclic AMP will be present, and no transcription of structural genes occurs. Let's understand this more clearly. When low level of glucose is present in the surrounding, high cyclic AMP will produce, which binds with CAP. Cyclic AMP is a hunger signal made by E. coli when glucose levels are low. Cyclic AMP binds with CAP protein, and changes its shape, so that it can bind to the DNA, and helps RNA polymerase to transcribe the genes. When the glucose is high, no cyclic AMP produces. In the absence of cyclic AMP, CAP cannot bind to DNA, so it cannot help the RNA polymerase to transcribe the genes. So low level of transcription, or we can say leaky level of expression, occurs. So, We have seen that for the activation of Lac Operon, two conditions are needed. First, glucose should be absent. Second, lactose should be present. Now, in the next slides, we will see four conditions. First condition, when glucose is present, but lactose is absent. When the glucose is high, no cyclic AMP will produce. In the absence of cyclic AMP, CAP cannot bind to DNA, so, it cannot help the RNA polymerase to transcribe the genes. In the absence of allolactose, LAC repressor will bind to operator, and inhibit the RNA polymerase to transcribe the genes. Second, when glucose and lactose both present. In the presence of glucose, cyclic AMP will be absent. In the absence of cyclic AMP, CAP cannot bind to DNA, so, it cannot help the RNA polymerase to transcribe the genes. Some of the lactose will convert into allolactose, and changes the shape of LAC repressor, which cannot bind to operator. Since, in the absence of CAP, RNA polymerase cannot transcribe, or if transcribes, then leaky level of transcription occurs. Third condition, glucose and lactose are both absent. In the absence of glucose, cyclic AMP will produce which binds to CAP protein and regulates it. CAP on binding the DNA sequence, helps the RNA polymerase to move forward. But in the absence of allolactose, LAC repressor binds with operator, and inhibits RNA polymerase to move forward. So, no transcription of LAC gene. Fourth condition, when glucose is absent, and lactose is present. In the absence of glucose, cyclic AMP will produce, which binds to CAP protein, and regulates it. CAP on binding the DNA sequence, helps the RNA polymerase, to move forward. In the presence of lactose, some of the lactose, will convert into allolactose, and changes the shape of LAC repressor, which can no longer bind to operator, so RNA polymerase can do the transcription. So, high level of LAC gene transcription occurs. This was a short introduction of Lac Operon. Thanks for watching. I hope you liked the video. Please like, share, and subscribe.